discussion part was done to some extent, Indian perspective, particularly in West Bengal. So I have to discuss this part also. So for the students, I will go slowly because to some extent, because this may be very new to you, because it is an animal disease. We normally discuss the human diseases most quite often, but it is a human disease, not a human disease. It is a solely bovine disease, and the cattle and buffaloes have been affected, and so it is related to veterinary having importance of veterinary science. So I'm Siddharthanaran Joardar. I did my master's and PhD from IVRI, and I'm just professor for 2015 in the Department of Veterinary Microbiology, West Bengal University of Animal and Fishery Sciences. Let us start. So in the overview, just I have to go for a virus structure, then economic impact to some extent, epidemiology, transmission, and it is having transboundary spread because it has got highly rampant migration from one country to another. I will show the pathway also. Very interesting story, and it's a very attractive for you also maybe. So I will touch that part, and we are seeing the signs of this particular the disease. Pathogenesis to some extent, I will discuss the clinical signs and symptoms, post-mortem lesions, to some extent diagnosis and treatment, and prevention control measures, very little because just I have to mention that this was actually African disease. It was there with the 90s, sorry. So, So when I was a teacher, 2000 around, and also in my student life, I never read this particular disease, lumpy skin disease, in our curriculum. It was written there, 1-2 lines, that lumpy skin disease is quite relevant to sheep pox, goat pox, and it is restricted to Africa, so it was not important to Indian perspective. Now, when it emerged in our country, particularly 2019, in the eastern side of the country, and we were affected, particularly West Bengal, Odisha first reported, that story I will tell. Odisha reported, but we could not report that one, although it came to West Bengal first, from probably Bangladesh, question mark is there, probably Bangladesh. So, anyway, so we were affected by economically also, and to some extent the problem was there with the animal husbandry practices that is going on, because we don't know the actual virus strain that is circulating in the country as also in West Bengal. The vectors responsible for this not known for particular incidences and outbreaks for this particular disease in Indian perspective. This has to be done, it's a new area. As also we have to go for vaccine preparation. Treatment is very difficult because it is a viral disease, and control measures, strategy to be taken for eradication, prevention and all. And so that's why it is very, very important at the time being. So it's a non-zoonotic disease, although, and also definitely vector-borne. To some extent, other sources are also there. I don't know what is going on there.
So do you have a mobile with you? Give it to me. Something.
No, it is not like that. Anyway, keep it. So let us go through. So it is a disease of pox viridae family. Pox is very known to you, and as you know, only one disease that eradicated from human, that is a smallpox. It is an orthopox family virus, but it is a Capripox virus disease. I will come to that side. The disease is characterized by fever, enlarged superficial lymph nodes. I will show the symptoms and lesions afterward, and limited range of vectors so far. It is going to affect the cattle and buffaloes only for the time being, as also some wild ruminants. Different names are there for in African countries, particularly Nidling and others are that Exanthema nodular bovis, and some other there, but lumpy skin disease is actually actual disease name, and it was first reported in Zambia in 1929, and then went to different different parts of the total African continent, maybe South Africa restricted to that, and certainly moved towards Egypt and towards Israel, towards other part that I will show. So it is kind of coming under Capri-Pox virus and subfamily Cordopoxvirinae and Poxviridae family. Under that, 8 numbers of genus are there. One of them is Capri-Pox. The brick-shaped virus, large subvirus. As you know, for AV-Pox and others that are very large size virus, maybe around 300 is a brick-shaped structure, and double-stranded DNA virus. It is a closely related to sheeppox and goatpox. Contains 30 structural and non-structural genes also. I'm going little fast because these are all known to you, maybe so to some extent, many months, the 35 days desiccation. Important is in the scab area it remains for long days. Necrotic nodules of the 33 days, and for height it is 18 days. So that is very important. And gets inactivated only 55 degree for 2 hours and 65 degree for whenever it is. We have kept it a virus in the minus 80, 10 years can be kept, and in culture if you keep it in culture form, at least 6 months you can keep for 4 degrees Celsius. Anyway, so some things are there, for some materials are there for susceptible, ether, chloroform, formalin. They can be used for controlling the virus lesions and all. And the economic it's affecting milk and meat and milk production and hide production. Milk production has also reproductive problem will be there for that. And draft power animals due to lameness, reproductive efficiency will be lost. And some are telling that agroterrorism there, I don't know, it has not so far. Anyway, prediction. And cattle or buffalo that I just mentioned, the target animals, calves go at the peak lactation show severe clinical signs and symptoms, and humans are resistant to the virus. So far, I don't know whether it will change its genomic pattern to attack human or not. Probably not. Because it is a RNA virus and it is not so much mutant virus, highly mutating virus, but local strains are available in different parts of the country, here also, and that is a problem for vaccine that I just mentioned. And mortality is low. That may be a problem for getting funds for me. Anyway, so it may be 1 to 3% mortality, maximum 5%, but morbidity may be up to 80%. Sometimes it goes beyond that. Normally 20 to 40% is the morbidity, and mortality is only 1 to 2. Mortality is there, 1 to 2%, particularly in calves. And in West Bengal it is going on now. Infection can be spread, non-vector transmission is also there, but mainly it is vector-borne disease, but non-vector transmission is also possible for direct contact. And our biting of different flies, mosquitoes and mites are there. Those are actually the vector. Buphailus, Rhipicephalus, Amblyomma mites, and only Stomoxys, the calcitrans, Culicoides and Culex mosquito. These are actually the vectors. Different species are there. I tell you, there is a deficiency of information for Indian perspective in Asia also. We have to go to identify, characterize those vectors, those are harboring the virus, and we have to establish a particular strain of virus that are going on to circulate in the locality, in our state, in our eastern part of the country. That has to be, several projects are going on for it. It is a very new idea, just I told. In Zambia, 1929 it started. In Egypt, it has touched 1988. 2006, the disease remarked to African Horn countries. And the first outbreak, 89 only, through Stomoxys calcitrans. If you see right hand side, I cannot show you because the problem is there. 2019, it has just touched India. A very beautiful picture was there, but I couldn't explain that one. Some problem is there with that. It has started from this side. You can cursor is not also coming. Okay. So this part that has started, Kazakhstan, Mongolia, and this route, virus is spreading through via the vectors. Very interesting. And the path we just see, sorry, it has come to this part. This part, first here it is coming to this side, then there is Bangladesh, West Bengal, Odisha, then again Nepal, and also China, and then touch to this part. That is Cambodia, Vietnam. That is not Vietnam. This is Cambodia, Thailand, and then Malaysia. To some extent, Indonesia. One after another years, 2021, 22, 23, 24, they are going to spread from southeastern states, southeastern countries. Now, outbreak first came in Odisha, but actually I know that it is in West Bengal, in Midnapur, and also the 24 Parganas north and south, where we could not report. Pressure was there, and I actually myself, my laboratory, anyway, I identified, isolated by some pressure was there. I couldn't reveal that one, report that one, but virus was there. 15 number of cases, third outbreak was reported. First published report was having no mortality, but 7.1 morbidity, close to South Africa. Odisha reported first. Whenever Odisha reported first, then I can go for, okay, that is there in our West Bengal. That, anyway, that challenges was also there. Tamil Nadu during this thing, 2020, report close related to Indian strain, Jharkhand, then Chhattisgarh, everywhere they know in India, from eastern side to southern side, and also northern side to some extent. If you go for a phylogenetic study, sequence study for an dendrogram, then you can see relatedness. And this relatedness is going to the Odisha is also related to West Bengal for one particular gene, and also another gene. It is a very relevant P32 gene. Here also you can see the closeness of Odisha and Bangladesh as also West Bengal. So development of viremia takes place first in the theoretical part, and localized in skin and develop inflammatory nodules, spread the virus mainly by monocyte driven viremia. Secondary sites are going to that, keratinocytes, tropism, hyperplasia, ballooning and degeneration of keratinocytes, formation of epidermal microvesicles, inflammation of cells, and coalescence of this thing. So, and there are several other pathogenesis. I'm not going to detail about this. It is not a place. Clinical sign, 2 to 5 weeks in natural cases, experimental in conditions for 7 to 14 days. And very characteristic features are there on the skin. So skin lesions are there just like pox naturally, but different from cowpox, buffalo pox. That has to be gone for differential diagnosis. And the illness begins and biphasic fever there, 2 lumps of nodules, 1 to 5 centimeters in diameter, develop within 2 to 3 days. Depression, anorexia, excessive salivation, ocular and nasal discharges, argalexia and emaciation. Height condition was not good. Milk production will be reduced. The animals cannot move further. That is the problem. And continuous high fever persists for 7 to 12 days. Serious depression, anorexia with several nodules with family uniform size all over the body. Nodular lesions, painful hyperemia. So many other these things there. Less milk production due to mastitis and abortion. It causes abortion also, and it is going to, the virus is going to affect the udder. Milk production will be dropped. So in that way, and also some reproductive problems will be there. That's why the problem will lead to certain high economic loss to the farmers. That is the main issue for human. Firm and slightly raised nodules are very typical lesions are there to identify. When lesions coalesce, large areas of raw tissue are exposed. The sloughed away lesions may create that is called inverted conical zone of necrosis. That is called Sitfast. It's very typical for this disease. That is very identifiable point. Diagnostic importance is so high for this. So, and also edematous and inflammatory swelling on the face, brisket, and one or more limbs may be very restricted. That's why they are going to have some restricted movement. deep ulcerative skin lesions, keratitis in some infected cows, pock lesions maybe so the pharynx, larynx throughout the alimentary tract, lesions of the respiratory tract followed by pneumonia. Internally, that is going towards the respiratory tract and causing pneumonia finally. There are so many ulcerative lesions for these post-mortem lesions are there for that respiratory digestive tract, and heart lesions are also there in endocardium, edema interalveolar and nodules are there in the lungs, trachea and gallbladder, and we have to go for differential diagnosis for this. Very easy is to perform polymerase chain reactions in primer specific to have some specific diagnosis. Clinical signs and that can be confused with other diseases maybe, but confirmatory diagnosis. Another one is isolation characterization and isolation of the virus from the normal way. The way we go for virus isolation and identification, and also PCR will be the confirmatory diagnosis for that. Virus nucleation test and indirect FAT and other serological tests can be also performed. As because it goes for an inflammatory phases, so naturally anti-inflammatory drugs, supportive treatments are there, but it is not so much important in case of cattle and buffalo for the farmers. If you go for a single-handed one in animal husbandry practice, if it is going for a large flock or rather large herd, then it is very important for treating the animals one by one. Individually, one or two animals, farmers may not afford also in that way. The treatment of LSD is costly, as well as the full recovery cannot be done and all. So restricted animal movement, hygiene control, vector control, I don't know, this is not there in our perspective. So actually no strategy is so far. Only strategy is to give a vaccine for goat pox virus, similar type of pox. I just mentioned once. This is a Capri pox virus genus. So Capri pox is having sheep pox, goat pox, and this lumpy virus, this lumpy skin disease. So we are trying to protect the animals from a specific disease, which is another animal species disease, that is goat pox, right? And live attenuated vaccine we are using for goat pox, the same Uttarkashi strain is recommended for Indian perspective. However, some of the local strains have been collected and trial of vaccines are vaccine preparation is going on, trials have been given, and some protection efficacy has also been estimated, but not commercially used so far. So we need a vaccine very much now for the homologous strain, Indian strain virus. We don't have any characterized virus, so we don't have that much characterized virus in our hand, so that we can prepare either cocktail or mixing with the other different type of local strains. Because India is a very vast country, pan-Indian perspective to be kept in mind. So we don't have that type of vaccine virus also in hand. That is the main issue. Sheep pox, goat pox vaccine can be used for LSD. Live attenuated vaccines. These vaccines are there in the African countries. Bovivax and that Wunderspoot biological products that they are preparing for needling strain. But very recently, ICAR, National Research Center in Equines and Hisar, with collaboration of Indian Veterinary Research Institute, they have developed one homogeneous live attenuated virus, LSD virus, that is called Lumpy Provac, but it has not been commercialized yet. So while discussing this thing, I should mention here for the research workers that science is something, administration is something. You cannot report all, but it is a sad part of the research. 2000 field veterinarians, my students, my colleagues, my classmates, they informed me that something is going on that is not in the field. It is not a cowpox type, something. When you taught in the class, one line was there, lumpy skin disease, but you never taught us, students telling like this. So we should go for identifying the virus and disease. It was 2019, July. I went there for isolating the virus for the lesions to be collected, isolation of virus to be done. And I thought in September last second, I got the virus, and it was lumpy skin disease virus. because of PCR identification was there, confirmation was there, I procured primers from the searching from the literature, and I got the virus. I informed the vice chancellor of our university. He told, "Chup, don't baat mat karna. I have to tell the animal husbandry department first, and then if they will tell yes, we can publish it, we can tell it to the others, then only we can do it." Somehow some reporters came to me, Vartaman and Eshamai. Reporters came me and asking, this is going on, this outbreak, this type of lesions are there in about from field veterinarian side. We got it, the information, whether the virus is for lumpy skin disease, something is there. Africa, Chormorok, Dokhinmuge, Shatadik, Guru Akrant, Udbigna Koldo. Lumpy skin disease, lumpy skin, they was asking like this. So I kept mum. So I told, yes, some indication is there, but this much of information is there for November 2019, before pandemic, 19. No advisory, nothing came from central side, central government side. But within 2-3 days, Odisha first reported the case, the outbreak was there. But I couldn't do anything. But my virus was in my lab. It is not still. It is with me. So I afterward, when all the things had happened, then I published that thing. I did the first isolation and identification, that molecular characterization of the virus. But I couldn't report. West Bengal couldn't report. Odisha reported first outbreak of Indian first report. And although the virus, I know the virus came from the Bangladesh side due to scrupulous crossing of cattle from this side to that side. This is the sad part of my last time when I was discussing the blue tongue virus for sheep and goat. Then I told that, okay, with the virus, my project was there, so we could develop one vaccine, diagnostics, and the characterized virus with us. We could report, but I couldn't report that one first. Anyway, so these are the references. I acknowledge our Honorable Vice Chancellor, West Bengal University Animal Fishery Sciences funding agencies and co-workers, particularly my students and fellows, and also definitely so I acknowledge the SOMA, the society, and Professor Bhattacharya, and also Dr. Sanyal, Professor Tyagi. I'm indebted to them, sir, for giving me a chance.
Wonderful lecture, sir. Wonderful.